Okay, we shall today we shall discuss about DNA topology, and as all of you know that uh, structure of the DNA that has been investigated by various scientists. We have also discussed in last lecture that there are certain models which were proposed by different scientists, and the most accepted model of DNA was James Watson and Francis Crick. Can you see the slides on your screen? Can you see
Yes,
the
sir.
slides? Okay, so DNA structure has been proposed, has been studied by many people, and Linus Pauling was one of them. Similarly, Maurice Wilkin and Rosalind Franklin. These were two other scientists, and James Watson and Francis Crick. And the famous model, which was accepted unanimously throughout the world, was proposed by James Watson and Francis Crick, which is also called as Watson and Crick model of DNA. As all of you know, familiar with important points of those Watson and Crick model. Their DNA is having double-stranded structure. They are interwound around each other in antiparallel form, and there is always a standard base pairing between the different bases of DNA. For example, A will always base pair with the T, and C will always base pair with the G. This is called Watson and Crick base pairing. So structure of when we talk about that was the model of the DNA, but in real sense, when we talk about the structure of the DNA is actually divided into four different levels on the basis of the structure of DNA. The first level is primary level of the DNA, the second is secondary level, then we have tertiary level, and we have a quaternary level. So these are the four levels of the DNA. When we talk about the primary structure is actually the linear sequence of the nucleotides that are linked together by phosphodiester bond, and this is what we can call as the linear sequence or the linear structure of the DNA, or it can also be called as primary structure of the DNA. And as far as a secondary structure of the DNA is concerned, this secondary structure is basically the interaction between the bases, in which the part of the strands they are bound to each other. So in double helical structure, the two strands of the DNA, they are held together by the hydrogen bonds. So that basically the helical structure in which the two strands of the DNA, they are interwound with each other in helix form, that will be called as secondary structure of the DNA. Then we have the tertiary structure of the DNA. In tertiary structure, there will be the location of the atoms in three-dimensional space. It is higher order structure than the secondary and primary structure, and in this tertiary structure, there is large-scale folding in DNA molecules, and as a result of which entire chain will be folded and a three-dimensional shape will be assumed, that is called as tertiary structure. There are four areas in which the structural form of the DNA can be different. One is handedness. Second important thing is length of the helix turn. Third one is number of the base pair per turn, and difference in the size
आप
between
स्लाइड
the. Yes.
स्लाइड्स मुझे नजर नहीं आ रही आपको शो नहीं हो रही
सर
स्लाइड
स्लाइड नंबर 1 ही शो हो रही है।
नंबर 1 ही शो हो रही है।
यस सर
यस सर।
अभी आपकी स्क्रीन पे मूव नहीं कर रही स्लाइड्स।
नो सर नो सर
ओके, लेट मी चेक।
ओके अब हो गया
हो रही है।
यस सर यस
ओके बाकी आपको आवाज आई है? आपने बाकी सुना है जो मैंने कहा है लेक्चर में?
यस सर बाकी सुना है
Yes,
पर
sir.
Okay, so there are 3 or 4 level of the DNA on the basis of the structure. One is primary structure, in which the linear sequences of the nucleotides, they are arranged together, they are linked together, and there is phosphodiester bond. Then we have secondary structure, just like that of the standard Watson and Crick model, in which the two DNA strands, they are interwound around each other, and they are forming a double helical structure. And when we talk about the tertiary structure, are any slides? Can you see the slide? Please unmute your microphone. आपको slide नजर आ रही है?
सर प्राइमरी स्ट्रक्चर और सेकेंडरी वाली शो हो रही है।
उसके बाद अगली नहीं हो रही?
Yes, sir.
टर्शियरी वाली नहीं हो रही?
नो सर।
ओके, प्लीज वेट।
Sir, show रही है।
अब हो रही है?
सर अब हो रही है।
Okay. So in case of tertiary structure, the atoms or the nucleotides which are present in the DNA, they are folded and they are assuming a three-dimensional shape. That is what we can call as. This is very higher order model, higher order shape, in which the atoms in the DNA or the nucleotides in the DNA, they are fold around each other, and they are forming a three-dimensional shape. So there are four areas or the four factors in which the DNA can be divided, on the basis of which the DNA can be divided into four different forms. Let me share this again. अब देखें आगे आ रही है? टर्शियरी से अगली स्लाइड आ रही है, नहीं आ रही?
नहीं आ रही सर, नहीं नजर आ रही।
ओके, अब आ रही है? ओके. The quaternary structure of the nucleic acid is similar to that of the protein quaternary structure, and quaternary structure, this is again very higher level of the organization of the nucleic acid. So here are certain level of the organization of the DNA. For example, this is primary structure. The primary structure of the protein is sequence of the amino acid linked together by peptide bond, forming a polypeptide. Here you can observe this is carbon and nitrogen bond. This is one amino acid. Similarly, this is another. So this is one peptide bond. This is second peptide bond. So in the primary structure, the amino acid are arranged in a linear form. In the similar way, in case of DNA, the nucleotides are also arranged in linear form, and in case of protein, there is peptide bond. In case of DNA, there will be phosphodiester bond between the linear nucleotide. This is the secondary structure in which the polypeptide, they can coil and they can form alpha helical structure. And similar is you can also assume this for the DNA, that in case of DNA, the two strand and the nucleotide, they will assume the helix structure, which is the Watson and Crick model of the DNA, a double helical structure. Then this is the tertiary structure. In the tertiary structure, the DNA molecule, they the protein, they may fold and they may form a higher order shape, which is three-dimensional structure, in which the regions of the secondary structures they form the tertiary structure. And then the last one is this is very a higher order organization, in which two or more polypeptide, they may interact and they may form a final protein, which is called as the tertiary structure of the DNA. So there are, on the basis of the various factors which we have previously discussed. For example, four areas. One is handedness. Handedness mean as we have discussed previously, that DNA is right-handed. This is called right-handed rule or handedness rule. So whether the DNA is right-handed or left-handed, the helix turn, what is the length of the single turn of a helix, and how many base pair are present in a single turn, and what is the difference between the major and minor group on the basis of these four parameters, we can differentiate the DNA into different forms. So here we will have different form of the DNA. We have A type DNA, B type DNA, cDNA, and zDNA. These are four different types of the DNA. So when we talk about the aDNA, this is aDNA. aDNA is a rare type of the structural conformation of the DNA, and this DNA can assume this shape under dehydrated condition. When there is scarcity of the water, when there is very less amount of the water is available, DNA will assume this shape, which is called aDNA. This shape of the DNA is called aDNA, and this apparently protects the DNA under extreme dry condition and desiccation of bacteria. When the bacteria are having a fear of desiccation due to the low availability of the water, bacterial DNA may assume this shape, which is called aDNA of shape. It is double-stranded, similar to bDNA, but it is shorter as compared to the bDNA. We will also study the bDNA in upcoming slide. Here you can see that this is more compact DNA, although it is more or less similar to aDNA, but as far as the packing of the base pair is concerned, as far as the size of the grooves that is concerned, it is more packed as compared to the bDNA. Similarly, double-strand is similar to bDNA but shorter and more compact, much wider than bDNA. The helix diameter of aDNA is 26. This will be I guess in nanometer or millimeter. The conformation of the glycosidic bond is in anti-form. I will tell you there are two form. One is anti-form, another is syn-form. Anti-form and syn-form is actually the binding of the nitrogenous base to the sugar that is having different conformational shape. उसको हम जो है वो एंटी फॉर्म और जो है वो उसको हम सिन फॉर्म करते हैं. ये मैं किसी और लेक्चर में आपको बताऊंगा व्हाट इज द डिफरेंस बिटवीन द एंटी फॉर्म एंड सिन फॉर्म. मेजर ग्रूव इज नैरो एंड डीप, and similarly minor groove is broad and shallow. Here you can observe that major groove is narrow and minor groove is broad and shallow. Similarly, aDNA is axial hole in the center. Axial hole. This is what we can call axial hole in the center. So these are some properties of the DNA that the DNA sometime it can assume a shape under specific condition when there is low availability of the water, and this shape is assumed by the bacterial DNA to prevent itself from desiccation, from drying due to the shortage of the water, and these are certain properties of this DNA. aDNA when we look at the bDNA, this is most uh, acceptable form of the DNA, and it is very much resembling to the universal double helical DNA, which is proposed by the Watson and Crick. So this is what we can call as bDNA. This is discovered by Watson and Crick. Most common and predominant form of the DNA is actually the bDNA. This is most common form of the DNA, most universal form of the DNA, and this structure resemble to that that structure which is proposed by Watson and Crick. It is double-stranded. bDNA has solid central core. There is no central hole as like of aDNA, so there is solid central core. Similarly, a major groove is wide and deep. In this case, when we talk about the major groove is wide and deep as compared to the minor groove. Similarly, minor groove is narrow and deep. Glycosidic bond is in anti-form conformation. Glycosidic bond is in anti-form. So as compared to the bDNA, this is in anti-form. Third type of the DNA, that is called zDNA. This is also the possible double helical structure of the DNA, and it is left-handed DNA. The bDNA is right-handed DNA, just like that of the normal DNA. This is called bDNA. I don't have the facility of the board, but simply I can show you. This is the handedness rule, in which the thumb of your hand. Can you see my hand? Can you see my hand?
Yes, sir.
No.
Okay, okay. The thumb of your hand is it should be along the axis of the DNA, and the helix should be in the direction of the fingers. This is called right-handed DNA. This is called right-handed DNA. You got my point that the axis of the DNA, this is the axis of the DNA, and the direction of your fingers, they should actually point the double helix or the helical structure of the DNA. This is what we can call it as right-handed DNA. So BDNA is actually the right-handed DNA. And when we talk about the zDNA, zDNA is left-handed DNA, and in which here the double strands are also in the zigzag fashion, but this is left-handed DNA. It has flat major groove. When we look at the major groove, there is no helical arrangement. It is having the flat major groove. Minor groove is to some extent that is narrow and deep, and glycosidic bond is anti and syn. It is anti for pyrimidine and syn for purine. This is the uh, glycosidic arrangement. Then we have another type of the DNA which is called as cDNA, C-form DNA. This is also double helical structure of the DNA, and this can be observed at certain conditions, certain environmental conditions. For example, when we have low humidity and the presence of certain ions, for example, lithium ion or magnesium ion, in the media we can have this shape. This is its shape is narrow. So normally we can have three shape. One is ADNA, BDNA, and ZDNA, and most commonly we always have the B form of the DNA. Z form and A form of the DNA, they form under rare condition, and C form of the DNA is also found under rare, extreme rare environmental condition. The C form of the DNA is may be assumed. So this is what we can call as different shape of the DNA. So shapes of the DNA can be differentiated on the basis of the handedness rule, whether the DNA is left-handed or right-handed, the diameter or the shape of their minor and major groove. These are two important points on the basis of which we can differentiate the DNA into various forms. When we talk about the topology, this is another term that is used. In case of topology, topology is basically the various aspect of the DNA structure that two strands are of the DNA, they are repeatedly intertwined with each other. ठीक है ना, ये जो डीएनए के जो दो स्ट्रैंड्स हैं, they are interwound with each other, and this aspect of the DNA, when this aspect of the DNA is discussed, that will be called as topology of the DNA. This will be called as topology of the DNA. A DNA segment constrained in such a way that its ends cannot rotate freely, it is called topological domain or topological constraint. I mean, the two strands of the DNA are interwound with each other, and no single strand can move freely on its either end due to intertwining of this strand with the complementary strand or with the opposite strand. So this condition is called topological domain or topological constraint. So here are various topological domain of the DNA. If we look at this DNA, this is circular DNA in which there is no free end. The DNA is in the circular form. Then we have the second type of the DNA. This is chromosomal DNA lobes. This is the arrangement of the DNA on the chromosome. Then we have C linear DNA, which is attached to the membrane. This is the linear DNA, and again it is not free to move at its end. Here you can see that the end of this DNA are actually linked with the membrane. This is linked with the membrane, and the last DNA is DNA which is attached or which is bound to the protein. As all of you know, the DNA is always present in associated with the histone protein in the nucleus. So this is the structure of the DNA, in which although the DNA is present in the linear form, but still it is bounded to the histone protein or any type of the protein. So all these shapes are actually representing the topological constraint or the topological domain in which the DNA is topologically constrained to move freely at its end. No end of the DNA is freely available. It is either the two ends are either bonded to each other, or the ends are bonded to any type of the protein or any type of the other membrane. So that is what we can call as the topological constraint. So DNA in its relaxed state, as I have told you, that it actually assumes the B configuration, which is most common, most accepted, most normal form of the DNA. It has the minimum energy and deviation. Any deviation can cause a change in the energy level. So when we talk about the prokaryotes, in case of prokaryotes, DNA is always circular, and it is this circularity leads to a lot of different properties of DNA. And that's why we can also call it as DNA is topologically constant. I mean, the two strands of the DNA are not available, freely available to move, and they are linked to with each other. So we can when we compare the eukaryotic DNA with the prokaryotic DNA, prokaryotic DNA is having more topological constraint. It is topologically more bound as compared to the eukaryotic DNA because of the shape of the prokaryotic DNA. That prokaryotic DNA is always having the circular shape, so the two ends of the DNA are unfree to move. They are linked with each other, so that's why prokaryotic DNA is topologically that is not free to move as compared to the eukaryotic DNA. Okay. Now, when we talk about the nucleotides, which are actually sugar, deoxyribose and phosphodiester linkage, they are same in all repeating nucleotides. So I think this slide is from previous lecture. We can omit. So number of the times one strand would have to pass through the other strand in order to form two strands to entirely separate from each other. This is called linking number. So as far as the topology is concerned, we have different terminologies that are used. One of them is linking number. Linking number mean the number of the time one strand, आपने देखा ना, आप दो रस्सियों को आपस में इस तरह से एक दूसरे के गिर्द क्या करें, उनको इंटरबाउंड करें, और दोबारा जब उनको अलग अलग करें तो जितनी दफ़ा एक रस्सी दूसरे से अलग होगी, दैट इज एक्चुअली कॉल्ड एज लिंकिंग नंबर. Or the number of the time one strand is interwound with the other strand, this is called linking number. It is always an integer. Linking number is the sum of the two geometric components. One is twist and other is writhe. We have two terminologies. One is twist and other is writhe. So linking number is always writhe number and plus twist number, and DL is what we can call DNA linking number is DNA writhe number plus DNA twist number. Twist क्या है? Twist is number of the helical turns of one DNA about other. ठीक है, जब हम उसको खोलना शुरू होंगे तो एक स्ट्रैंड जहां जहां से दूसरे स्ट्रैंड के साथ लिंक हुआ है, वो जितने टर्न खुलेंगे उसको हम लिंकिंग नंबर कहते हैं. और ट्विस्ट नंबर क्या है कि द नंबर ऑफ द टाइम वन डीएनए स्ट्रैंड इज इंटरवाउंड अराउंड द अदर, दिस इज कॉल्ड ट्विस्ट नंबर. उसको हम क्या कहते हैं? ट्विस्ट नंबर कहते हैं. एंड व्हेन वी टॉक अबाउट द रिथ नंबर or wrist, this is not wrist, this is writhe number. This is basically the supercoiling of the DNA. When the DNA is supercoiled, it is number of the turn of the axis of the double-stranded helix in space, that is actually called as writhe number. So here we can have the clear idea of the linking number, of the twist number, and of the writhe number. So here linking number is 23. So what do we mean by the twist number? Twist number mean 25 times this DNA is interwound with other strand. For example, this is 1, this is 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and similarly so on. So here here you can observe that this is actually the twist number, that one strand of the DNA is interwound at the other strand, and the total number of the point where one strand is interwound to the other strand, that is called twist number. So twist number in this case is 25, and writhe number is 2. Writhe number is 2, so there is negative super helix. What do we mean by writhe number? Writhe number mean that this strand is itself, then it is interwound. This is what we can call as twist number. So writhe number is 2, so we will have the linking number is twist number minus writhe number, so linking number in this case is 25, and this is right-handed DNA, where the writhe number is in negative form, that is why this is right-handed DNA. So, there are special enzyme, as I have told you that the DNA is topologically constrained, and it is unable to move at its free end, ये अपने एंड के ऊपर जो है वो मूव नहीं कर सकता. तो अब इसकी ये जो टोपोलॉजिकल कंस्ट्रेंट है, इसको फिर रिमूव करने के लिए क्या होता है, देयर आर सर्टेन एंजाइम्स व्हिच आर प्रेजेंट इन द डीएनए और इन द बॉडी, एंड दोज एंजाइम्स दे एक्चुअली remove these topological constraints. Those enzymes are called topoisomerases. Enzyme को हम क्या कहते हैं? Those are called topoisomerases. They actually change the DNA topology. They are present in eukaryotes as well as in prokaryotes. ये prokaryotes में भी मौजूद हैं और ये यूकैरियोट्स में भी जो है मौजूद है. ठीक है, और अगर हम इनमें देखें, if you silence the genes of the topoisomerases in the eukaryotes, major processes for example replication, transcription, that will also be stopped. Because when we talk about the eukaryotic DNA, as all of you know that eukaryotic DNA is present in the chromatin form. क्रोमेटिन फॉर्म मीन कि वो टोपोलॉजिकली बहुत जो है वो कंडेंस है, वो कंस्ट्रेंट में है. तो ट्रांसक्रिप्शन आगे हम डिस्कस करेंगे, रेप्लिकेशन और ट्रांसक्रिप्शन. Transcription and replication and transcription are only possible when this topologically compact DNA or constrained DNA is free from those topological constraints and it assume a linear shape. तो जब तक वो linear shape assume नहीं करेगा, DNA में जो है वो replication का process भी नहीं होगा, transcription का process भी नहीं होगा. So that's why to remove those topological constraints, the DNA is having specialized type of the enzymes in the body. Those enzymes are actually called as DNA topoisomerases, और वो सारे के सारे जो टोपोलॉजिकल कंस्ट्रेंट्स हैं उनको जो है वो वहाँ से रिमूव करते हैं. So this is what we can call as DNA is having the source of genetic information, and it is having various structure and various conformational shapes, or the various conformational structure of the DNA, which we have already discussed. For example, we have A DNA, we have B DNA, we have Z DNA, and we have C DNA. So this is all about today. Okay, you may unmute your microphone and you may ask the question. If you have any question, please. Okay.
So ये linking number एक.
Okay, please wait. Okay, let me clear this. For example, here is linking number. Can you see my hand? ये मेरे हाथ में एक तस्बीह है, ठीक है. अगर इस तस्बीह को अभी आपको नजर आ रही है?
हाँ सर.
नहीं नजर आ रही? क्या आ रही है?
सर थोड़ा ऊपर करें ना, नजर नहीं आ रही.
आ रही है?
यस सर. वीडियो ही ऑफ की हुई है.
जी नहीं,
मुझे नजर नहीं आ रहा. चलो ठीक से बताइए.
मेरी वीडियो तो ऑन है, मेरा कैमरा तो ऑन है.
नेट कनेक्शन पुअर था तो ऑफ कर दीजिए.
ओके ओके, ये देखें, ये जो तस्बीह की ये दोनों साइड्स हैं, व्हेन दे आर इंटरवाउंड विद ईच अदर, दिस इज कॉल्ड ट्विस्ट। इसको हम क्या कहेंगे? ट्विस्ट कहेंगे, ठीक है ना। और अगर इसी एक ट्विस्ट में हम फर्दर इसमें जो है वो कॉइलिंग करें, दिस इज कॉल्ड रिथ। इसको हम क्या कहेंगे? दिस इज कॉल्ड रिथ, ठीक है ना। और ये जो एक टर्न बना है बिटवीन दीज टू स्ट्रैंड, ये दो स्ट्रैंड्स के दरमियान जो एक टर्न बना है, इसको हम क्या कहेंगे? दिस इज कॉल्ड ट्विस्ट नंबर। और जब हम इसको खोलेंगे तो दिस इज व्हाट वी कैन कॉल, सो ट्विस्ट एंड लिंक आर सेम इन द सेंस, इसमें नंबर ऑफ द टाइम वन स्ट्रैंड इज इंटरबाउंड अराउंड द ईच अदर, ठीक है। उसको हम क्या कहते हैं? दिस इज कॉल्ड ट्विस्ट नंबर, ठीक है। और अगर इसी ये जो लिंक्ड है या ये इंटरबाउंड एरिया है, इसमें हम फर्दर सुपर कॉइलिंग करेंगे तो दिस विल बी कॉल्ड एज रिथ नंबर। इसको हम क्या कहेंगे? रिथ नंबर कहेंगे, ठीक है। सो हियर इन दिस एग्जाम्पल और इन दिस स्ट्रक्चर, ये सारे के सारे क्या है? दीज आर द ट्विस्ट नंबर। ये क्या? ये ट्विस्ट नंबर है, ठीक है ना। इनको हम काउंट करते जाएंगे, यानी जहां जहां पर ये उसके साथ जो है वो लिंक कर रहा है, दिस इज ट्विस्ट नंबर। और जब हम इसको खोलेंगे तो उसको हम जो है फिर लिंकिंग नंबर भी कह सकते हैं, कि नंबर ऑफ द टाइम वन स्ट्रैंड इज इंटरबाउंड अराउंड द अदर स्ट्रैंड, दैट इज कॉल्ड लिंकिंग नंबर, ठीक है ना। और रिथ नंबर क्या है? कि आप उसी में फर्दर जो है वो क्या कर दें, उसी में फर्दर दो जो है, लेट्स सपोज ये हमने एक एक ले लिया, दिस इज सिंगल वन। ये पहले वाला जो है ये हमने एक सिंगल एरिया ले लिया, इसी में फर्दर ये कॉइलिंग होके दो इसमें और हमने बना दिए, तो ये अब क्या कहलाएंगे? दिस विल बी कॉल्ड एज रिथ नंबर। इसको हम क्या कहेंगे? रिथ नंबर कहेंगे। क्लियर है
यस सर।
जी? एनी अदर क्वेश्चन? एनी अदर क्वेश्चन प्लीज? कोई नहीं है क्वेश्चन? समझ आ गई आप सब लोगों को? ओके, कैन यू सी दिस स्लाइड नाउ? अब आपको स्लाइड नजर आ
सर
रही
जो
है?
आपने कहा था,
जी जी।
सर जो
जी
आपने कहा था राइट हैंडेड डीएनए,
जी। राइट हैंडेड
सर
डीएनए
उसका
मीन, मैंने आपको बताया ना, दिस इज माय हैंड। क्या आपको नजर आ रहा है?
यस सर।
कैन यू सी माय हैंड? ये जो है, दिस इज द एक्सिस ऑफ द डीएनए, ठीक है ना। ये डीएनए का एक्सिस है, यानी वो दरमियान से दो स्ट्रैंड के दरमियान जो आप लाइन लगाएंगे, उसको हम क्या कहते हैं? दिस इज द
यस
एक्सिस,
सर।
ठीक है। तो ये जो आपका अंगूठा है, ये डीएनए के एक्सिस को जाहिर करेगा, और ये जो आपकी फिंगर्स हैं, दे विल पॉइंट द न्यूक्लियोटाइड, यानी इस तरफ उसका हेलेक्स होगा, उस तरफ क्या होगा? इसका हेलिकल टर्न होगा। दिस इज कॉल्ड राइट हैंडेड। इसको हम क्या कहते हैं? राइट हैंडेड कहते हैं। लेफ्ट हैंडेड के आप इसी को उल्टा कर दें, ठीक है ना, कि आपका ये जो अंगूठा है ये उसके एक्सिस को जाहिर कर रहा है, और इस तरफ जो है वो उसकी क्या है? उसके जो है वो टर्न है या हेलेक्स है, दैट विल बी कॉल्ड एज लेफ्ट हैंडेड डीएनए। तो नॉर्मली जो बी डीएनए है वो क्या है? दैट इज इन द राइट हैंडेड कन्फॉर्मेशन। किसमें है? राइट हैंडेड कन्फॉर्मेशन में डीएनए।
यस सर।
ओके, अभी आपकी स्क्रीन पे कौन सी स्लाइड चल रही है? लास्ट वाली, कंक्लूजन वाली?
यस सर।
ठीक है, अब कौन सी चल रही है? अभी वो फिगर वाली स्लाइड है आपकी स्क्रीन पे। कैन यू हियर मी? अभी आपकी स्क्रीन पे कौन सी स्लाइड है?
सर्कल वाली स्लाइड है सर।
ओके, फिगर वाली स्लाइड है ना जी। प्लीज स्टे कनेक्टेड, लेट मी अपलोड योर अटेंडेंस। अच्छा, एक और चेक करें आयशा, ये आप में से कोई
जी
दूसरे को रिमूव करके देखे, क्या आप रिमूव कर सकते हैं? अभी चेक
यस
करें
सर,
किसी
कर
से।
सकते हैं।
अच्छा, अभी वेट करें, मैं भी बताता हूँ। अब चेक करें आशा, कैन यू रिमूव अदर स्टूडेंट? किसी को रिमूव करें?
सर रिमूव करूं?
अभी रिमूव करें।